TREM2 is an important receptor that regulates immune cells such as microglia and macrophages. Mutations in TREM2 are associated with neurodegenerative diseases such as Alzheimer's disease. A recent study published in Biology investigated how TREM2 influences cytokine expression in macrophages. Macrophages were treated with TREM2. Multiplex human cytokine ELISA kit was used to detect levels of tumor necrosis factor alpha, monocyte chemotactic and activating factor, interleukin 6, interleukin 1 alpha, interferon gamma, interleukin 1 beta, interleukin 8, and granulocyte macrophage colony stimulating factor. The researchers found that TREM2 induces the expression of inflammatory cytokines early, but promotes anti-inflammatory cytokines at later stages. This occurs primarily via the MAPK-JNK signaling pathway. Additionally, TREM2 has differential effects on cytokine expression on different macrophages based on their polarization. These findings suggest that TREM2 plays a dynamic and context-dependent role in immune regulation, with implications for neurodegenerative disease mechanisms like Alzheimer's disease. Anogen's multiplex human cytokine ELISA kit was used in this study to assess cell inflammatory cytokine expressions. Anogen supports researchers in immunotherapy and neurodegenerative diseases by providing hundreds of high-quality antibodies and ELISA kits. For more information, please visit Anogen's website at www.anogen.ca.